So good morning to everybody. I'm so very thankful for coming and participating in our session, which is devoted to our priority research area number 1, computational oncology and personalized medicine. We have a keynote speaker who kindly accepted the invitation to give a talk during our session. It's Professor Philip Lambin from Maastricht University. He describes himself, if you go for the website, as the clinician, scientist, and radiation oncologist. And when you look at his research, it's exactly what it says. He's a pioneer in translational research that focuses on tumor hypoxia, genetically modified bacteria for cancer treatment, and the thing which is the most interesting for us for the moment is the decision support systems. So he got a PhD in molecular radiation biology, and he's a professor at the University of Maastricht, and that's the precision medicine area, and then at the University of Eindhoven, and it's about the functional imaging. So if you look at his, let's say, scientific achievements, he is the co-author of more than 600 peer-reviewed papers, and they got 56,000 citations. If you go through the Google Scholar, his Hirsch index is 114. I've checked it today morning. And IT index, you know, the number of patients who got at least 10 citations is 515. He is the co-author of more than 20 patents that are closed or ongoing, and the promoter or co-promoter of more than 66 completed PhD, let's say, procedures. And what is very crucial for today's talk, he is the co-inventor of the idea for the radiomics, which is very important in imaging informatics, and today's talk is devoted to focuses on that area. So, Philippe, thank you so very much for accepting the invitation, and the floor is yours.
Well, thank you very much, Joanna, for these nice introductions and also for the invitation. So indeed, today I'm going to speak about radiomics. So what does it mean? Well, you could guess, because there is rads for radiology, so imaging, and omics meaning lots of data, right? But the idea is to transform medical imaging, which is often seen as sort of a qualitative sort of type of data, in a quantitative mineable data that can be used to improve outcome of patients. So here, my disclosures. So I'm a co-inventor of the company Radiomics and a few patents licensed. But if we make a step back, so we have to realize that the medical doctors now is confronted to a large amount of data with genomics, imaging, clinical data. Not only that, but also a lot of different treatments. So you could think, well, that's good, right? But it's not necessarily, because the human cognitive capacity of a normal, well-educated doctor is 5. So he can handle 5 variables, and often it's only 2, black and white. But if he's well-educated, 5. So we could argue that by adding a lot of information, we basically confuse the doctor. And that's why it's important to build a decision support system. And also I want to point out something which people don't like to talk too much about, is that healthcare is a very chaotic, error-prone process. For example, in the US, which is one of the examples, right, there was about a little bit less than 500,000 of people who die from medical errors. So that's more or less equivalent to 2 jumbo jets full of passengers crashing every day. And in Europe, it's the same. And it's probably even worse after the COVID, when we have sort of exhaust personnel, etc. So it is a very chaotic process, and we need to improve that. And so, like a lot of people, I believe that the future lies in human-machine collaborations. We won't replace human by machines, but we can make them collaborate so that they are more efficient. And in this machine, we need to look at imaging, because imaging is used everywhere in medicine, right? Even in psychology and psychiatry now, they use MRI of the brain. And how to handle imaging and how to innovate in imaging, there are basically three ways. First one is to convince the management of the hospitals to invest in a very expensive machine, for example, MR PET. Second is to use new contrast agent, especially in PETs, sort of nanobodies, etc. And the third method is to use the imaging we already have, but try to extract more information out of it by using automated software, by using artificial intelligence. And that's really what we are going to discuss now. So radio mix hypothesis is simple, is that at the moment, still imaging, when you look at the report of in the hospitals, they are mainly qualitative reports. There are very few numbers, sometimes the largest dimensions of the tumor. And what we want to do is to move towards a quantitative image analysis with lots of numbers, errors bars, etc. And that's actually a revolution. That sounds obvious, but that's, I tell you, it's a real revolution. And why should we do that? Well, I like to show this very simple image. This is a very sort of a simple image with only 36 pixels, right? Black and white. And if I ask you which one of those two squares is the darkest, most of you will say, well, that's a very stupid question, because it's quite obvious that the one on the top is darkest. But actually, if I tell you no, they have exactly the same density. Most of you will not believe me, because especially in medicine, we have been educated to believe what we see. But what we forget is that what we see is not a reality. What we see is processed by your brain, by the cortex of your brain. If I do this, perhaps you will believe me, this is exactly the same density because there is a shadow which gives optical illusions. So that's one of the problems. We don't see the reality with our eyes, and what we see is not quantitative. So we could also, that's another method which has been tried, you can educate pigeons to read medical images. Because you know that pigeons, they always find their ways, even if it's cloudy. They have a very good sort of geographical vision. You could educate them to look at pathological slides of mammographies. And when it's a tumor, they have to tick on the screen and they receive some food behind. And believe it or not, the female pigeons are usually much better than the male pigeons. The male pigeons are much easily distracted, less focused. If they work in team, they're working better. No, we won't do that. That's too complicated. And also they get tired very quickly, by the way, the pigeons. So we want to use machines. And let me give you here another example. It's the letter A with different pixels of different grays, right? And then for each pixels, we can put a number. For example, when it's black, it's 0. When it's white, it's 1, and intermediate. So we basically have different gray level. It's not the 50 shades of gray, no, it's 1,000 shades of gray. And from this right matrix with different gray levels, we can basically analyze it. We can look at tumor image intensity, for example, how many black and white do we have, and grays, to just look at the numbers. Then in the second, we could look at the shape. For example, here it was the letter A, but we could have the letter B, so the shape of the tumors as well. Then we could look at textures. How many similar or dissimilar pixels do we have next to each other in 3 dimensions? And then we can also do multi-scale filtering. For example, we can decide to divide all the pixels, the numbers in the periphery by 2, and multiply the one in the center by 10, whatever. We just invent a filter, and then you redo this exercise. And then there are more complex features, fractal features, radial gradient that I won't discuss here, but the possibility to create new features is nearly infinite. So we end up with thousands of features from this matrix of gray level. And just to give you a concrete example, here on the top panel, we have lung tumors. You could see it's smaller than the bottom one, but it's very heterogeneous. You can see it by eyes. So this translates by a peak of gray level, which is very broad. So you have a lot of heterogeneity in the gray level, while in the bottom panel, we have a larger tumor, which is very homogeneous, and this translates by a narrow peak of gray level. So just by looking at this, that's a very simple matrix. You see a big difference. And you have to think in terms of biology. The biology of those two tumors must be totally different to create such a different phenotype. You must have different clones, heterogeneity in the tumor microenvironment, etc., in the top tumor. So basically, by looking at this simple matrix, you also get a matrix, quantitative matrix of what's going on at the biological level. There are also some other features like mesh-based data. While you look at microarchitecture, you can look at shape, semantic location. And then you can also invent new features. For example, here it's a French group, they invented, when the patient have metastasis, they invented the longest distance between two metastases. And they found that when the distance was big, the spread in the body of the cancer was bigger, it has a very strong prognostic value. That's a very straightforward features. And then I would like to say a few words on one of the other, I would say, co-inventors of radiomics, which is Bob Gillis, which passed away, unfortunately, a few months ago. And he made these famous statements: images are more than the picture, they are data, right? That's a very key message from this talk. And then he tells the story that he was very frustrated to see that he's a biologist, that he knew that tumors were very heterogeneous. And he said they were frustrated to see that there was one single value used to describe a tumor, like SUV max, K-trans. So he knew that was not true. And indeed, when we look at intratumor heterogeneity, if you look at different slices of the same tumor, you could see that's, for example, a different slice of H&E stained tissue from the same tumor. You see this massive heterogeneity. So you can understand intuitively that if you take a good biopsy taken randomly in very heterogeneous tumors, it will not necessarily be representative of the whole tumor. So that's intra-tumor heterogeneity. But we also have intra-patient heterogeneity, because most of the cancer patients have multiple lesions, and the lesion in the brain and in the liver are not the same. So we won't be able to make biopsies of all those lesions. What we can do, though, is try to make a phenotyping of all the images by all the lesions by images. So that's, for example, an attempt to phenotype the multiple lung metastases of these patients with different colors. That's something you can do with imaging. So radiomics can lead to this concept of the virtual biopsies. You have the real biopsy. We take one piece of tissue somewhere, but you cannot do that for lesions. But you can do, what you can do with imaging is sort of describe all the lesion quantitatively. I will pass that. And then Bob Gillis, in 2008, he spent a sabbatical in Maastricht, and then we discussed a lot, in cafe, restaurants, etc. It is that time that we came with this concept of radiomics by using all we have, like we do in genomics, to quantify, to just describe imaging. And we published these first papers in 2012, which is highly cited and really starts the field. And now you could see on PubMed there was an exponential increase of papers citing this concept of radiomics. I will pass that. But it's a nice concept. We'll use standard imaging, but we have to also take into account two things. Is that the data quality. If we have bad images, we won't get a lot out of it. And also the big data paradox, namely that the more data we have, the sure we fool ourselves, unless we use a very rigorous methodology. That's something I want to describe further, how to now capture those information from images. And data quality is certainly important, and the sentence, you are what you eat, is also valid for radiomics. You need good quality of images, homogeneous, And if they are not homogeneous, you need way to correct for that. And you know that in data quality, we have the Facebook data quality very low. Then you have the bank quality very high, luckily for us. And the medical data, really, it has to be at this end as well, high quality. So in a radiomics, there are basically two families. The first one, handcrafted radiomics, which I've just described you with intense shape texture, right? And there, the process is simple. You segment a lesion of interest. It can be a tumor, it can be a lung, it can be a liver. It doesn't have to be cancer, obviously. Then you will extract the features with a software. There are good open source software like P-Radiomics to do that. You make a radiomics analysis with machine learning. And then what you hope is to generate information that can be used for medical application like diagnostic, is it a cancer or not, prognostic, predictive radiomics signatures. And if we make a step back, this is what we produced in around a little bit before 2012. So we had about, on the y-axis, about 400 patients of lung cancer, all treated by radiotherapy. So we took the CT simulation. And then on the x-axis, we had about 600 radiomics features. And we had no idea how to analyze that. We had a big Excel file with lots of features. We asked a statistician some help, and he said, try to do unsupervised clustering. So basically, the computer tried to put together the patients which are similar. You could see there are a little bit sort of three clusters on the right side, in the middle, and on the left side. And then in the right side, there are the adenocarcinoma. In the middle, there are squamous cell carcinomas. And in the left, small cell lung cancer. So basically, a CT, an analysis of a CT without contrast was able to guess pathology. And in that time, that was sort of a, mind-blowing, because we realized that there is biological information in simple anatomical CT, which was not of use. So we thought, okay, if we can see pathology, perhaps we can also see gene mutation, we can guess the prognosis, etc. So that's really how it started. And that led to the second hypothesis of radiomics, that we all agree that our phenotype is determined by our genes, that the basic dogma of biology. But what was not obvious is that we also hypothesized that we could imputate what's going on the gene level, molecular level, based on the analysis of anatomical images. And how do we do that? Well, let's say that we have on the right side imaging that we analyze. We generate image features. On the left side, we have genomic analysis, and we try to map features and genes. And then we try to correlate really biology with imaging. One example is human papillomavirus, which is a cause of head and neck cancer. It's increasing. And we know that HPV plus patients, which are typically determined by P16 assay or directly by measuring HPV in the tumor, HPV plus patients have a better prognosis. But we can easily determine whether a patient is HPV plus or minus based on a radiomics analysis on CT. And if we look at survival of the patients with a HPV plus diagnosis based on in blue on radiomics or in red by the classical molecular assays, we can see that the prognostic values are the same. So we can do as well with radiomics than with molecular assays to predict HPV and survival. This is another example of lung cancers. So here we also analyzed radiomics, and we did some genomic analysis. And then we use a more complex method called normalized enrichment score. And then there are sort of modules. And you could see in red on the top bottom that we found that there are 3 modules which are linked to survival and that's the modules 9, 12, and 2, which are basically P63 and the immune system. And then when we analyze independently those biopsies of patients with a radiomic score high for 12, 9, and 2, we could see that they had more lymphocytes, CD8 plus cells in brown on the left sides. So we could sort of validate independently with another method, a molecular assays, that was a link between radiomic scores, lymphocyte in the tumors, and survival. Something that can also be done is to try to go, when you have large tumors, you have, for example, CT PETs. You have some areas within the tumors with a high SUV. So you can analyze separately the high SUV areas of the tumors and the low SUV areas. And then you can also better definitions, a better analysis of the lesions. But this is only possible if the tumor is rather big, because if you have very small nodules, then you don't have enough information anymore if you try to divide the lesions in different parts. Okay, now let's move to the second big family of radiomic, which is deep learning. So deep learning, in contrast to handcrafted radiomics, who have predefined features, hey, you don't have predefined features. You learn by experience, you learn by trial and errors. So you need typically big data and GPU farm, and then the computer will try everything until it finds signatures which can answer your question. For example, differentiating cancer from non-cancer, survivors from non-survivors. And but those two methods, which were presented a few years ago as competing, can actually collaborate with each other. They are complementary. But let's make a step back how deep learning started. It started with this Imagenet competition, where basically you have thousands of images. You can see on the right side, you have dogs, you have cats, etc. There was a competition for the software which was able to recognize the most images. And the performance was not great. There was more than 25% mistake until in 2012, there was the first solution to have less than 25% mistakes. And that was the first method using deep learning. And now the performance of this new software is excellent and is with errors less than 5%. And they all use deep learning. And deep learning can also be used now, for example, that's published in Nature, to look for nodules, cancerous nodules in screening CTs of patients who have smoked. And you could see the blue line compared to the cross. The cross are the performance of radiologists. The blue line is better than radiologists and also much faster. So how those methods can collaborate? The first obvious things, for example, is that you can use deep learning, a variation of deep learning, UNet, to automatically segment the, because in the early stage of radiomics, we had to do that manually. That was very time consuming. So now we can do that automatically, and then you do the whole pipeline. This means that those analyses can occur automatically in the backgrounds of the PACS of the hospitals. But there are other methods, and for example, here is a case where we analyze patients with idiopathic lung fibrosis. We did handcrafted radiomics and deep learning. And then you could see here that when we do ensemble learning with handcrafted radiomics and deep learning, the performance in terms of accuracy much higher, 0.85, than when you use handcrafted radiomics and deep learning separately. And then, by the way, you could see that the performance of the clinician is 66, is the lowest of all the analyses. So you show that those two methods can collaborate. It is also another approach, is to look when those two methods, which are totally different, agrees, the one there is a consensus. For example, here, deep learning and handcraft radiomics agrees in a little bit more than 60% of the case. And then when you look at the specificity and accuracy of the subgroup for which both method agrees, then it's very high. It's higher than 90%. So the sub-conclusion is that those two methods of radiomics, handcraft and deep, can collaborate in at least three ways: segmentation, automatic segmentation with deep learning, and then handcraft radiomics analysis. Second, ensemble learning. And third, looking at consensus. So you could say to the radiologist, look, for this subpopulation, we are pretty sure because the two methods agrees. For this other subpopulation where there is no consensus, you should spend a little bit more time. The problem of deep learning is that you need often a lot of data, and often in the medical field, we don't have a lot of data. So there are different methods. The first one is to use synthetic data. This is one of the first painting created by a generative adversarial network. It has been sold nearly 500,000 at Christie. And this is, the movie King's Lions. At the left side was when my kids were small, and that's the last version of the movie. So it's very impressive what they can do. So if we can do that, we can also generate synthetic mammographies. And that is very helpful to actually help the training. Another method is distributed learning. So you have, you analyze data from different hospitals, but the data stays in the hospital. So you don't centralize the image in one computer. So you have a sort of learning module. So you go to hospital 1, hospital 2, hospital 3, and then make several iterations. So the data never leave the hospital, so we don't have privacy issue and all this nightmare with legal departments, etc. And the third method, which is probably the most promising, is called transfer learning. So you have a deep learning, let's say, to recognize cars, but you can reuse this deep learning algorithm, at least the first layers, to segment anatomy on CTs. And the first layers are typically the same, and then you gain a lot of time. You need much, much less patients to train the model. Because the first steps like edge detection, color histogram, pixel segmentation, they are the same if you want to recognize scars or segment anatomy. Okay, let's now go to the clinical utilities. Why is it useful? That for three reasons: diagnosis, follow-up. Let's say to first tumor volume. Tumor volume is a very simple radiomics features, but not yet used in the clinic. But we know everybody understands it. Big tumors are worse than small tumors. It's very simple. So we just published papers in Nature Communications, and I will summarize it to you. So we know that when we ask doctors to segment tumors, there is a huge variation. For example, here we have a very simple tumors controls by 5 experienced radiation oncologists, and you have different controls. So we hypothesized that we could do that automatically, much faster and better than doctors. We could save time for the radiologist, save time for the radiation oncologist, increase reproducibility, have less noise, and better evaluate tumor response. So we train with a lot of images from different hospitals to have a large variability and different kernels. And then we knew that the frequency domain was very, very different from those images. For example, here, 2 examples from 2 images, the frequency domain is different. So we found a way to harmonize that. And then, like a mathematical formula, we first try to simplify the image by extracting the lung and then normalizing the images because they were coming from different CTs, different centers, and then detect with whether there was a lesion, and if there was a lesion, segment it and reconstruct it in 3 dimensions. So that's what we built, and then you end up with this type of control. So in red is the predicted by the computers, and in blue is done manually. It's called the ground truth, but you could argue whether it's really the ground truth because if you ask different doctors, you will have different answers. And so you could see here, you can measure that by DICE index, you have different image. And basically you end up with the predicted contours, the surface, but also the volume of those tumors. And then we decided to do an in silico trials to evaluate whether this method is better than the doctors. What is an in silico trials? Well, it's based on the principle that there are also level of evidence for biomarkers in medicine, like the level of evidence for treatments. And according to the definition, if you have a prospective trial with as primary objective the biomarkers, then and you validate your biomarker, you have level evidence 1. So we thought this is actually also valid for AI. And that should also work if we do an in silico trial. And then you say, what is an in silico trial? Well, an in silico trial is like a real clinical trials in the sense that it's pre-registered, you have protocol, you have sample size calculation, you have several endpoints, main endpoints, secondary endpoints. But the difference is that you use digital material of real patient, of synthetic digital material. It's much faster and it's cheaper. So this is the platform of a digital trial. So for example, that's when I participate. I said, okay, I'm a radiologist. My name is Santa Claus. And then I have the same patient, which is contoured twice, and once by the machine, the AI, once by experienced radiologist. And I have to say which one I prefer. I can play with the window level, look in 3 dimensions, and I have to do that 100 times. I did that, and I found that in 57 times, I prefer the contour made by the AI. And then when we ask lots of doctors and data scientists to do that, they tend to prefer the AI-based contour. But on the right side, the medical doctors and radiologists, they tend to prefer much more often the AI-based contour than the manual contour. So that's a point, right? It's not a decisive point, but it's a point. Then we look at the time needed to do the contour. Here we ask sort of 7 doctors, and it took about a few minutes to 15 minutes, while for the AI it took a few seconds to do the contour. And that's important because you could, gain efficiencies, especially that we are lacking radiologists nowadays. So we could say, okay, not much better actually at the moment, but certainly much faster than doctors. Then we look at intra-variabilities. We have the same doctors to do the same control at 3 months interval. And then you could see that the average DICE for the doctors on the same patient was only 86%. But sometimes it was 0. Sometimes it could even not recognize the nodules delineated 3 months ago. While the DICE, the reproducibility for the AI was 100%. That's a good argument because this means that the noise will be much less than with doctors. And then there's a final argument. We hypothesize that if the AI is better than the doctor control, the prognostic value of the volume designed by the AI should be better than the prognostic value of the volume designed by doctors. That's what we found. When we look at the prognostic value, we have a data set of lung cancer patients treated by chemoradiotherapy. When it's based, we split the survival based on the automated contour on the right side, the hazard ratio is higher and the p-value is lower, is better. And we did that on another data set of patients treated by stereotactic radiotherapy, same thing. The differential effect of the AI volume is higher. So this leads to us to think that it's not only faster, more reproducible, but also better. Then let's give you another example for diagnostic. So we always try to start from an unmet clinical need. So we need to speak with radiologists. And here the problem was the part solid nodules on CT. They are very difficult to evaluate for radiologists. So we want to develop radiomic signatures to help radiologists to differentiate malignant, non-malignant part solid nodules. And we decide to analyze the radiomics of the whole things, on the left side, then only the solid, the ground glass opacity, second, third STV, only the solid part, and fourth, on the right side, the whole things plus the margins around the nodules. And to make a long story short, we found that the two in the middle, namely the ground glass opacity and the solid part analyzed separately, gave the best discrimination between malignant and non-malignant. So that could be very useful for doctors. Then how to treat? Here I will refer to a European study which has been done on head and neck cancers with a part which was retrospective, a part which was prospective. So that's one of few. And then basically we have retrospective 600 patients and prospective more than 140. And we have mainly the CT. And then when we look at the retrospective data on the left side, you could see we could discriminate three groups: green, yellow, red, based on purely radiomic signatures. And we could sort of validate that on the right side. But when we use a multi-omics approach, which is the FUTURES, which is basically radiomics plus clinical plus biological factors like P16, hemoglobin level, then the differential between the curve was higher. And it was actually even better in the prospective part. So you could see on the right side, the green curve is nearly flat. So those patients have very, very good prognosis. So we could really consider to do dose de-escalation to make the treatment less intensive for this green group, because people have tried only based on P16, it didn't work. P16 only is not strong enough. But perhaps this combination of radiomics and multi-omics approach would work to decrease the intensity of the treatments. So if we look at AI, I think like all techniques, you have a sort of a peak of inflated expectation. Then people realize that there are some problems, it's the AI winter, and then there's a plateau of productivity. I think we are there about here, because initially people realized there are a lot of problems, and now we are trying to solve those problems. And when you look at the European Commissions, there are requirement for AI. They want something fair, explainable, lineage. They want to know where is this software coming from, and robust. That's what they call trusted AI, TAI. And I will speak a little bit about explainability, because I realize that doctors will not use an AI they don't understand. And I don't know if some of you have seen the movie with Tom Cruise of Minority Report, where basically people were put in prison for a crime they have not yet committed, they were likely to commit, they were put in prison preventively. So that's unacceptable ethically, of course. So because the doctors want to understand if there is a statement, are you sure of that? When do you fail? Can I really trust you? So the next step really is to have an explanation interface, an explainable model, and then have the doctors asking questions and getting confidence in the statement of the AI. So there are a lot of days of interpretability methods to try to understand the AI radiomics, and I won't go into details. You can look at the review there. Basically, there are two families. There are post-hoc interpretability. So you make a model, then you explain it afterwards. And there are most interesting one, interpretable by design. So they are made to be interpretable. So to give you some examples, this is AI that we use to look for COVID pneumopathy. And here you have in color on the top panel, you have in color the voxels which are the most informative to make the diagnosis of COVID or not. And in a way, you are reassured that the voxels correspond to the abnormalities on CTs. Because sometimes the heat map is, for example, on the bone, but then you don't believe it, right? There's no way that bone image would help you to differentiate COVID pneumopathy. So that's one way. That's a very easy way, very standard. There are another way, for example, where the anatomical priors for glioblastomas, so for example, so where the doctors know out of experience that when the midline of the brain is shifted, then it has a meaning, and it has to be taken into account. So those type of anatomical priors based on knowledge are taken into account in the AI outcome. But there are many more methods. Second challenge is the issue of image acquisitions, because we have a lot of different CT, different protocols. For example, convolution kernel. Here, a phantom with 2 different convolution kernels, same CT. You could see the difference. There is no way that we could extract the same information from those 2 images. This is another example. The same patient with images in 2 different centers. Look at the difference of quality. It's very, 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 very different. So there are different ways to look at it. One of the papers here published this year, so we try to look at what are the factors which are the most important, because to have exactly the same images, that won't be possible. And basically, we concluded that the most important parameters are the slide thickness, and the convolution kernel. So that's the most important. And they explain a lot of the variability. So I will pass that because I'm a little bit running out of time. So there are also methods, statistical method, like here, COMBAT, which is one of the classical methods. When you have images coming from 2 different centers, you look at the variability with sort of a statistical trick inspired by genomic analysis. You try to move the data point together to decrease the variability. That's called the COMBAT methods. You have also other methods like style transfers. It's like, you know, in Word, you go from Arial to Times New Romans at once. There are also methods to do the same with images, right? Like you transform an image in sort of impressionist painting. And the most promising are GANs, Generative Adversarial Network, where you try to basically transform an image to another image of reference by using generative adversarial network. But the challenge is that's easy to do. The challenge is to do that without losing information about the biology of the tissue. So a lot of research ongoing on the speed. I'm pretty sure in a few years we'll have the solution for that. Okay, so last point, data quality. I insist on that, but analysis method is very important as well. And I think the key element really is when you do radiomic studies is always to have an internal validation. So you have the tripod classification, you probably know it, but you can find it easily. Type 3 is that when the same research group use an external validation data set. And type 4 is the best, is when another group will validate a published signatures on a totally different data set. So that's absolutely really needed. Personally, if there is no external validation, I always reject the paper. And then we also develop a score, radiomics quality score, which is available on the website radiomics.world. And I won't go into too much details, but we basically look at all the steps which are important, which can have an impact on the outcome, and we sort of deconstruct the analysis in all small steps, and we give a score for each steps. And this radiomics quality score is used more and more by reviewers for grants papers. And you can, if you go to the website, you can try your papers and test your papers with this score. So what I believe is that the clinician who do not use AI will be replaced by the clinician who does. So I don't say that AI will replace the clinician. That's no way. And there's some example in chest where actually sometimes when the AIs collaborate with chest professionals, they are beating AI. So there's something to explore there. And then that radiomics, which is basically quantitative image analysis, exists in two families, handcraft radiomics and deep learning, allow you to extract information, biological information, clinical information for diagnose, a treatment decision, or follow-up. And for CT, the most important factors are convolution kernel, slice thickness, and pixel spacing. And I think I'm convinced that radiomics will help busy clinicians to make decisions, faster decisions. And the first application will be segmentation and diagnostic questions. So there are a few issues to be solved, the quality control, and that's why we have this radiomics quality score, the explainability. There are different methods now to do that, image harmonization, especially when images are coming from different CTs, different MR. And also, I didn't mention that prospective validations. You can do that in silico, actually. And for the people who are interested, we organize every year for now, the last 5 years, international course in June. In the morning, we have lectures by top speakers coming from everywhere in the world. And in the afternoon, we have hands-on exercise with 2 tracks, one for data scientists, one for clinicians. So if you're interested, visit that website. And I would like to acknowledge my colleagues, and then I'm happy to answer any questions and eventually to collaborate. Thank you very much.
Thank you. Thank you so very much. I'm playing the discussion, and you can ask the questions. Those who are online, you have an opportunity to unmute yourself and also to put the camera on. So if we have some questions from the online public, or if there is anybody from here, from the room, and then you ask the question. So in the meantime, if there is no question, I've got 2 comments or question, let's say that way. The first one is about the problem of lack of the data or images for the deep learning systems. You mentioned that we can go for the GANs to do the synthetic images. But as you remember, you also mentioned that there is a huge variety or inter-patient variability. So if we go for the GANs, then still we are limited to the original data that feed the system. And we will get more images, but will not cover the problem of that variability, inter-patient variability. So,
yeah. So, there are 2 parts to these questions. Indeed, we cannot rely only on synthetic image, but what you can do if you have a data set of images, you can make a synthetic one which has the same features, the same characteristic than original one.
Yep.
The advantage of synthetic one is you can share it. You can put it on the website, there's no problem. Yep. But you have to ensure that they have the same, I would say, normal distribution of features, of key features than the original one. What you can also do is that, for example, if you have a breast cancer, and then you have a rather rare subtype like lobular breast cancer. It's not so frequent. And the AI has difficulties to recognize lobular cancers because it's not in the training data set, there were not a lot of images. Mm-hmm. Then what you can do is take the lobular, and then you make 10,000 copies with some variations of that lobular.
Okay. Mm-hmm.
And then you do a new training with this sort of rare subtype, which has been enlarged. And then when you do that and you test it on an external validation, you see that the performance of your software increase. So basically, it's a little bit like a student, who is at the university, is good in everything except in maths. What he's going to do is going to do lots of maths, He's perhaps going to take a private tutor to do maths, and then he will succeed. So you could do the same with AI. You could just boost the training on certain subtype, and if you don't have the real images, you take a synthetic image with the same characteristic than the original one.
Yeah, that's it. I agree. Okay, so we let's say make the systems more robust against the imbalance of the training data set. Yeah. Okay. Do we have some comments from the audience, from the students, or some question? Okay, we've got the one.
Okay. Yeah. Thank you very much. Thank you very much, Professor, for a very interesting
I have a
presentation.
question.
Okay. Just.
I have a question. If student want to train his or herself by this technique, so is it possible that
There's the online student, we cannot hear you. Oh, yes.
is it
now we can.
possible that they can do by online?
Sorry, I didn't understand the question. Could you repeat it?
I want to ask that if a student want to train herself by this technique to using this radiomics for imaging of different types of cancers, tumor cells. So is it possible that they can do some workshops or trainings online, or it's compulsory to come physically and you train them over there at your institute or something?
I'm not so understood it, but I mean, you can train online, yes, especially with the method of distributed learning, where you have, for example, different hospitals who keep their images in the packs, and you don't centralize it. So you just have to install a module allowing federated distributed learning, and then you can learn online. So basically, without bringing physically the images in your computer. I'm not sure that was the question, but that's what I was understanding. Otherwise, perhaps if I didn't understand well, just rephrase the question, please.
Okay. I think it's, in case you can write and contact Professor directly, and then you can...
Yeah. Don't hesitate. Here's my email. Don't hesitate to send me your questions, and I will answer it.
Okay, thank you. So now we have Professor Piętka willing to ask the question.
Okay, yeah, thank you very much. So once again, Professor, thank you very much for an interesting presentation, very wide your experience. I have just a question about training. You've said that you're using images coming from various places, various databases. Now my question is, in those places, they are using different CTs, third generation, fourth generation, with different also reconstruction algorithms. And that is why we deal not only with the problem with the spatial resolution differences, but as well as the problems with reconstruction, where you also get just different images. You can see the same, but as you've shown in the very beginning, that you can see two rectangles, one is white, one is black, and as a matter of fact, we see the same. It might be the same problem in here. Using different reconstruction algorithms, as a matter of fact, you get different images. Does it impact somehow the teaching problem?
So you're totally right. That's I mean, also what I said in the conclusion number 3 is that there are 3 factors which for CT, for MRI is even more complicated.
Yeah, that's right.
But for CT is convolution kernel, slice thickness, pixel spacing. Okay, so in ideal world, you would like this to be similar. So you can do like in a clinical trial. In clinical trials, there are inclusion and exclusion criteria. If the patient don't fulfill the inclusion criteria, they are not included in the analysis. You can do the same. You can say, you're not obliged to take all the images. If there is an image suddenly with 5 millimeter slice thickness, you could reject it. If the convolution kernel is very different, you can say, no, sorry, I cannot do that. I cannot analyze that. So that's one option. And that's easier in prospective trials because you can request the images to be taken in a certain way. Another option is to do post-processing harmonization. That's the combat methods. You accept different images, and then you look at the distribution of features, and if you have 2 normal distribution, you try to harmonize that. That's a method which works a little okay, but it's not great. And then the third method, which I think is the future, is to do some pre-processing harmonization. So basically, you will harmonize them by using generative adversarial network. You will try too much. Now nowadays, it is possible to transform, it's done in routine in several centers, to make CT images for MRI. For example, that's used a lot in prostate cancer. They do an MRI because it's a better soft tissue resolutions, and then they transform in a CT for dosimetry and for planning. So if you can do that, you can also transform a CT from CT1 to an image of a CT2. So these are, I would say, in summary, the three options you have.
Thank you. Okay, and on chat, I've got one last question because we are running out of time. So the question is that you mentioned about the PET, about the MRIs, about the CTs. These are maybe more or less invasive, still invasive techniques. And how about the ultrasonographs, which is, let's say, treated as the one being the less invasive one? Do you see any possibility of using radiomics in that area?
Yes, definitely. We just have now papers submitted where we use liver ecography. It is absolutely possible. The same methods applies. So you have handcrafted radiomics, deep learning. I mean, the extra difficulty is that you have is very much dependent of the radiologist. The inclination of the sound, etc. So there is some variability, but you can correct that. But yes, it can be used actually for, in theory, any medical imaging. For example, we use also for OCT, optical coherence tomography, which is used for eyes and for dermatology. We use it for eyes fundus. We use it for pathology. So in theory, any images is possible. I would say the most difficult until now in my experience was PET, because PET, there's a lot of variability between centers, and that's very difficult to correct. But otherwise, it is possible, but it is more difficult.
Thank you so very much. Okay, I've got one question from the audience. Professor, could you please come here? Yes.
Good afternoon already. Thank you for a fantastic lecture, Evarist Kac, Faculty of Biomedical Engineering. Professor, you mentioned during your lecture just once and showed just one slide concerning frequency analysis. Do you think that more advanced method like higher order spectra at the level of feature extraction would help a little bit to be more precise and more accurate with the processing of further data? Thank you.
Yes, indeed. I think it's an interesting method. I just try to go back, and there are different methods actually. Yes, here is a sort of in these slides you have a sort of an overview of all the methods, right? And then something also attractive is also to use the raw data, right? Because at the moment, what we see are highly processed data, and there's big companies, Siemens, GE, etc., which spend a lot of time to ensure that the image looks good for the human eye. But by doing so, they eliminate a lot of information. So I think the future will be also to go to the raw data and perhaps do a special processing for AI, because it could be that the processing for AI is different than the processing for the human eye. The radiologists want to see something different than the eye. So I very much believe in these different methods, GAN, raw data, etc., which all occur before you start to really analyze the image.
Thank you. So thank you so very much for your lecture and the audience for questions. It was a pleasure to have you here, and we're looking for the future cooperation. And now we proceed to the second speaker. It's about the application of the methods we've already mentioned for the COVID-19 system for the diagnosis. So I am right now...
Thank you very much.
Thank you. So I am giving the chair to Professor Piętka. Thank you.
Okay. Thank you very much. Now it's my pleasure to introduce the second presentation, which will be given by Dr. Michał Marczyk. He is an assistant professor at the Department of Data Science and Engineering at our university. His research interest is in the bioinformatics, data analysis, and development of prediction models in cancer research. Doctor, the floor is yours.
Hello, everyone. Thank you so much for this nice introduction. Thank you for inviting me here. It's a great pleasure for me to present you the results of the project called CIRCA, which was under supervision of Professor Polańska, which was run in a group of the few specialists. And with this system, we aimed to develop a tool and an online platform for COVID-19 and pneumonia, other pneumonia detection, using one of the simplest medical imaging modalities, which are chest X-ray images. Oh, it's not working. Oh, yeah. Thank you. It's working right now. So, from our perspective and from the previous presentation, we already know that now the machine learning, AI, deep learning tools are really important in the clinical research, diagnostics, and can help or support clinicians to have faster decision, faster prognosis. So we already know it from different diseases. And within the medical imaging modalities, we often use chest X-rays, computed tomographies, or MRIs, or PET, or others. But the chest radiographies actually are the cheapest, are the easiest to access, and what is important in the times of big rush in hospitals, it can be done portably by the patient bed by a single radiographer. So that is why when we entered this pandemic era in 2020, there was many different tools, the COVID-19 diagnostic tools that appeared fast in the literature. So in 2020, it was about 200 publications. In 2021, twice as them. So now we have hundreds or even thousands of different tools. So it's great, right? We'll see. So now we are in the era where we have a lot of data, where we have a lot of computational resources. So naturally, almost all of these models are based on deep learning. And since we analyze medical images, they are based on convolutional neural networks. But still, even that we have many solutions, some time after, there appeared papers or reviews that try to validate these solutions. And of course, many of different problems were found with these AI systems. In this work, the authors use some kind of explainable AI tools to show on the actual images where are the places in the image that the deep learning model used to make a decision about the patient. And they found out that almost all of the systems made the decision based on artifacts, rather what's important for medical pathologists. So they used the method called Saliency Maps, which on this slide is this red dots show you the regions based on which the system tells you that this is COVID-19 or not. And it turns out that even you can see that it's totally wrong, right? Because you need to look on the lung area and then decide if there is some differences, some pathologies. But this system mostly decides on the region outside the lung. And the problem is not in the researchers, but in the data, right? Because we know that for deep learning, we need a lot of data. We need a lot of good quality data, which we need a lot of representative data. But since the start of the pandemic, the amount of data was limited. Also, the databases were specific for a disease. So we had pneumonia patients from one database, COVID patients from other database, normal healthy patients from another database. So there were different biases that were introduced. Also, it was found that in one of these classes, the patients were, for example, younger than in the COVID-19 class, right? So many biases caused training of different, maybe wrong models. So our first solution to this problem was to define this region of interest, which will be similar to what radiologists look. So we wanted from this raw image to extract only the lung area. So we did it in a few steps, starting from some basic preprocessing to standardized images to normalize the contrast of these images. And then we used the UNet network to find this lung segmentation here. And at last, as the post-processing, we use the convex hull method, because in some more advanced cases, more advanced COVID-19 cases, it turns out that it was not possible to have the full region of the lung. So this will increase the area, which is important for a classifier. In another paper, they describe 62 different models developed in 2020. They found out that half of them were developed on the public data sets, other on the private data, so there was no chance to validate that models. Since most of these papers were produced really fast, they used pre-trained networks, so networks trained on the, for example, natural images or other kinds of images. And the amount of data that was used to develop these models was really low. So only six papers were trained on over 1,000 images. And the reported accuracy in all of these methods was 80% or more. So still, if we know that it is hard to create a good model, they showed that it could be produced. And the conclusion of this work was that none of these currently proposed methods is product ready to introduce to a clinic. And the main reasons are similar. So we have small size of data sets, especially for COVID-19 class. We observe a huge variability in these data sets from different countries, from different sources. And there is no collaboration between data analysts and clinicians to validate if the prognosis of the deep learning models, of the AI models, are similar to what radiologists will tell. And one of the data set that was used in most of these papers is called COVID-X database. And it was a great initiative, because this is an online database established in 2020, in which individual researchers and medical doctors can upload their images. So it's a great way to get a lot of data really fast. But then you have multiple problems with quality, with different biases of this data. And also the problem was that all of this data were stored in the graphic formats, like compressed JPG format. Also, people only uploaded the COVID-19 patients, but the other pneumonia patients were collected from totally different database. That is why we decided to create a probably better database, which was based on the images taken from different medical centers in Poland. In this database, we took the X-rays, chest X-rays of healthy people, people with COVID-19, and people with other pneumonia. We asked doctors to use the DICOM standard to save these images to keep as high resolution as possible. And we also tried to select these medical partners to have different medical facilities around the Poland that use different scanners and different methods. So the solution to this another problem. Our solution is to create the more representative training set. To do that, we also spend a lot of time to clean this data because data quality is really important. So from more than 6,000 images that we got, only 4,800 were ready to be used to create a model. We also cleaned the COVID-X and database, and we found another clinical data set, which is called AI for COVID, which stores the COVID-19 patients' images from 3 different hospitals in Italy. Then, after that, we tried to create a nice scheme to split the data into the training data, which are here, into the holdout set for testing these methods. And also, we gathered some independent data sets, which comes from the Spanish hospitals. And since we had a not very well balanced data set for training, we used 2 other databases or data sets to balance our data. So we used the pneumonia patients from another database, NIH dataset, and we also used the solution already mentioned today. So we designed GAN-based networks to artificially create images, CXR images of COVID-19 patients. And here are just an example of data that were artificially generated. So they have the ribs. These data are after pre-processing, so after selecting only lung region. Okay, so then when we have all this data, we were looking for some method to visualize all data and see how different are they from each other. And in the bioinformatics, when you work with numeric data, TSNE or UMAP method starts to be really popular because from the thousands of features that they are able to visualize, to create only 2 features, which could be easily visualized. So one point here is one image. But when you run it on images, people mostly just vectorize this image and create a UMAP on this vectorized image. We propose different methods, and we used 2 different solutions. First, we calculated radiomic features on the images and then put radiomic features into UMAP. Or second, we trained the supervised deep neural network and take some features from last layers of these networks. So in these 2 different solutions, we were able to have better representation, 2D representation of our full data. So here the colors show you these different 3 classes. So blue is pneumonia patients, which are located on the top left, the normal patients, which are located on the top right, and COVID-19 patients, which are located here on the bottom. So these classes are not separating very well. So it's not that the problem is really easy, and based on these features, they are totally separated. Also, we find this island here, which looks that have some images with different features. And when we look deeper and compare these 2 databases, COVID-X and POL-COVID, we saw that most of these patients are from the COVID-X datasets, and all of them are from the COVID-19 class. So because we don't have any points here. And then we try to see if this is just an artifact or this is just a different pathology of COVID-19. So we, again, use different radiomic features to differentiate between the images that are here and the rest of them. We use different information from this DICOM images. But it turns out that the problem is more simpler. So it turns out that here we color-coded the original resolution of the image that was inputted into deep learning model. So we use as an input, we use the resolution of 512 pixels to 512 pixels, but some of these pictures were smaller, and they need to be upsampled. So it turns out that all these pictures had lower resolution. This is the artifact caused by upsampling. And other researchers did the same. When we try to downsample the images, all images to 256 by 256 and rerun the creating of this UMAT, it turns out that this is not a problem. They are not looking as an outliers. So we didn't want to remove all of this data, so we decided to create a new model. We create a model for the super resolution model to increase the resolution of original images. It was based on ERS-GAN architecture that was developed for other types of data. We train the images, train the model by downsampling the images with two different resolutions, 256 by 256 and 128 by 128. And it turns out that it was a good idea because from all these outlying samples, 41% were properly corrected by the model. So some of these patients that were located here goes to the region which is closer to other images. And the last thing, the last problem that we noticed is that even if you have three classes, healthy, normal, healthy, pneumonia, and COVID-19, still there is some heterogeneity within each of this class. So we decided to quantify, to group these patients into some subtypes, and we feed the two-dimensional Gaussian mixture model to UMAP representation of each class, and introduce the class calls N1, N2, and 3. So it's normal 1 to 3, pneumonia 1 to 3, and COVID-19 1 to 3. And we believe, based on just a location, the UMAT, that N1, P1, and C1 are the most representative for each class, but N3, P3, or C3 probably will be much harder to quantify. The performance will be lower. And finally, to increase the robustness of acquiring all these images and also to give this tool online, publicly free for everyone, we create this online portal where each of you, which goes to the circa.aei.polsl.pl, can input the data and can see your diagnosis and can see in which subtype your score was. So the final pipeline was created based on this smaller blocks that I already described, but since it is an online tool, we found some problems that people try to upload different images, that lung X-rays. So we also added some tools. So we added the tool to verify these images that are uploaded, and this verification was based on first running the segmentation process, lung segmentation process, and using some shape analysis, we can say if this is CXR on other, and we also create the SVM-based classifier to separate these images, and here are the exemplary results. So when we got the negative class for this SVM training, so the correct images, we can see that the generated lung mask is okay, and also the classifier says the same. But when we have, for example, another type of medical image, not chest X-ray, you can see that these masks are really bad. And when we have different images or the same modality X-ray, but it's that lungs, also the generated masks are not good. We tried different photos like human faces, some natural examples, and it worked nice. Okay, and the last thing is, which I didn't mention, is that after all this pre-processing step, after preparation of all this data, so definition of this region of interest, we tried to create a good prediction model. And we tried first from the model based only on radiomic features, but then we add another system. We add a second model based on inception V3 architecture. This is a convolutional neural network. and we use it on this pre-processed images. And we also used another dense neural network based on radiomic features, which were first pre-processed using effect size analysis. And based on these two classifiers, we use the construct a decision tree to aggregate the prediction outcomes from these two methods and give us the final decision. Okay, and as you might remember from this data splitting scheme, we try to make a few steps of validation. So first, during training, we had the training and validation data, and we also ran this cross-validation results using 5-fold cross-validation, and here are mean and standard deviations from these results. And when you look per different class, it's about 90% accuracy. The sensitivity is a little bit lower than specificity, and since this is a clinical test, we trained the models so that the negative predictive value is really, really high, and positive predictive value could be a little bit lower. When we took the holdout test set, so the part of images from this free databases that were not used during model training, but still are similar to this training data by some features, we can see that results are still good. So the accuracy is from 81% to 92%. Still, a negative predictive value is higher. And we can also see that the results are a little bit better for this COVID-X database than our clinical data set. So clinical data set was harder to predict. But when you look on the subtypes of these three classes, we explain somehow which patients are harder to predict. So I told you that C1, N1, and P1 are the most representative, right? N3, P3, C3 are least representative. And we can see this on these results, right? Look on the balanced accuracy for N3, P3, and C3, and compare it to C1, C2. So if you have disease-specific symptoms, we can already see it on this UMAP, on the subtypes, and it's easy to predict that you are in a given class. If your symptoms are not specific, if you are similar, if your image is similar to the image of patients from different class, the model also works not very well. And these were results for POL-COVID database. So this holdout set on POL-COVID, the holdout set on COVID-X. So here, when you are in N1 or P1, you have the perfect accuracy. And also on this another clinical data set from Italian hospitals, where this accuracy in C1 and C2 was really high, 97%. But in C3, we have a little bit lower sensitivity, which drives the accuracy down. And the last important part, we found, or they introduced the data sets from the Spanish hospitals, and we use it for independent testing. But first, we try to visualize using this UMAT where are located these patients. And when we look on the classes, we see that in our cases also, normals were on the left, COVID were here, and pneumonia were on the bottom right. So, but here we have some problems with pneumonia patients. They are located all over the image. They are not grouping here like in our data sets. So we said that maybe we will have some problems with classification of pneumonia patients, and it was true, right? So for the pneumonia class and this data set, the balance accuracy was a little bit lower, but still C1, C2, COVID-19 class, which have this patients with typical and atypical COVID-19 symptoms, we got more than 90% of balanced accuracy, high PPV, and high NPV. And the last comparison that we did is for this independent data set, they asked radiologists to group this data into 4 categories. If on the image you see the typical appearance, atypical, indeterminate, or you say that even that the test was positive, this is the patient which is negative for any signs of pneumonia. And when you look here on this table, 76% of COVID-19 patients, because only COVID-19 patients are here, that were classified by our method into normal classes, were also scored by radiologists as negative for pneumonia. On the other hand, when you look on the typical appearance, they are mostly grouped in the C1 or C2 COVID-19 subtype. Okay, so to conclude, we first try to find what is the problem of current solutions, which was mostly the really poor representative data set. So that's why we gathered different clinically relevant data sets and using this data splitting scheme, try to create a more robust model, the model which will generalize better. And we also developed multiple tools to clean this data, to select only important, only clinically relevant regions of this image. Looking on these results, we show that this CIRCA system shows a really good performance to distinguish COVID-19 patients during training, during initial validation, and also on these independent test sets. We also provide these subtypes to show this, to reveal this clinical heterogeneity, to explain why the results in some subgroups of class could be worse or not. And finally, we have this online portal that we maintain all the time, and we provide easy and public available access to support the diagnosis of COVID-19. At last, I want to acknowledge all the people that were involved in this project. So the head was Professor Polańska, and the rest of us were manually create the manual annotations, cleaning the data, creating the different models, do the validation, and creating the portal. And of course, there will be no results with or no good models with a good infrastructure. So Russ thanks is for the infrastructure that we have on our department. Thank you.
Thank you very much for an interesting presentation. And now the paper is open for discussion. Are there questions from the audience or from the online participants? Okay, maybe meanwhile, I'm going to ask a question. You're talking about cleaning the data, having 6,000 images and cleaning it down to 4,000. Did you do it purely manually, or you had at least a semi-automated method to do it?
Yeah, it was really hard to do it manually. So we tried to use, for example, this UMAP or calculate some distances between these images, for example, to remove the duplicated images. We look on the DICOM information to see if the image is from the same site of the patients, right? So we try to use this different information to initially filter some data, and then the last needs to be manually.
So if you had an image that was flipped, for example, left to right, you have just removed it from the data? Or
No. If there were some images that were able to correct, like flipping,
okay.
Yeah, we correct that.
Okay. Any other question? If not, then I have maybe one more question. Who divided those classes into 3 groups? You said that N1 to 3, P1 to 3. Who divided it? How was it divided into those 3 groups within each class?
Yeah, so we use this unsupervised method. We use this 2D GMM modeling to let the machine find the division.
Okay, thank you very much. Any questions from the audience or from the online?
From online, there's no question.
Okay, since we don't have and I don't see any questions, thank you very much. Thank you very much to both presenters. Thank you very much to the audience. Thank you for the questions. And now this session is closed.
Ladies and gentlemen, allow me to thank